So we've got two different groups of people here. Right now you've got the Grand Challenge Explorations, and the proposal you're looking at is the Grand Challenge Explorations proposal, and that's been funded. I believe with the diagnostics, it's slightly different. But what we're trying to say is this structure may not be exactly how it works for another RFP, but the concept of how to tell a story, how to make sure your budget makes sense with the scope of work you're working on, that's going to be the same regardless of which project or which RFP you're applying for. And it's about how you tell that story may differ, and what you tell in each story might differ. But the connection point and how you engage your readers, in many cases, will be the same regardless of which RFP you're applying for. So we're trying to really help you pull out the major themes. In this case, the GCE proposal is the one that we're able to share with you today. I think there was a question here.
Yes. So in your objective number one, I guess it's going to be a small molecule screen. Why you take the approach to say activity 2 and activity number 3? What is the difference between both of them? Because from what I read here is you say, I'm going to screen 5,000 drugs. And it seems to be that and then you start, if necessary, in activity number 3, if necessary, we're prepared to raise the stakes. That means, like, if I don't find anything, I will go to 525. I mean, it's just, why you divide it in two and not make a single one?
Right. It's, again, the way I think about this. I think in all of the proposals, I'm going to take some time later to go through some things that I've highlighted. And what I've highlighted are these constant worries in the back of my mind. If you just show what you think is worth doing, and you don't tell the reviewer what you're worried about, all the different ways that you think your project might fail, then you might be at risk of appearing naive, right? And so what you want to do is convey this idea that you've thought about this, and you know what the funder wants, right? Bill and Melinda Gates want to eliminate malaria parasites. And so there are a lot of projects out there where people will claim to impress somebody by either numbers of compounds that they can screen, or how fast they can screen, or how many different proteins they've developed for a screen. You see all versions of this. What we were trying to do was to create a plan where we've thought about all the different options, and we're trying to streamline everything into a manageable size project, stay as close as possible to deliverables that the funder cares about, and then go to the more complicated stuff if necessary. That was the overall thinking. So if in this particular example, we've now got a system where we can have parasites in a well that are not resistant to this compound in a single well of a 96 well plate. We're trying to come up with a scheme where we challenge each well with a different molecule over a certain amount of time, and then we score for whether the parasite successfully developed resistance to the test compound. So there's a test compound in the well, and then there is this inhibitor that's going to block the ability of the parasite to develop resistance to the original challenge. That's a fairly sophisticated assay, and it's not like looking for a flash of fluorescent light. So you don't want to do this with, or propose to do this with 500,000 molecules as the first thing you do. And these molecules have their own liabilities and all kinds of problems. You're just setting yourself up for all kinds of smashing on the head by different reviewers. So we set up 2 stages. First, to work with about 5,000 drug-like compounds. And this is grantsmanship, but it's also what I believe in. I want to be able to get up while I'm still alive and say, we help with this particular problem in malaria treatment. I want to start with 5,000 compounds that have gone through humans, that have proven to be safe. Many of them can be taken for long periods of time. If we can find a hit within these molecules, then that would help the project move towards where the funders want to be very fast. Now somebody can say, well, screen with just 5,000 molecules, what are you talking about? So then there is this argument that if you're targeting a specific enzyme or a particular protein, and it has a very rigid pocket, and you have to be able to find the right binding environment within that pocket, you might need 100,000 or 200,000 molecules to find one that will hit. And we've done things like that for specific targets. The argument that I make in the proposal is that we're attacking a process. The process has multiple players. any of these players hit by any compound would stop the process. So if you play the statistics games of blue marbles and red marbles and all this stuff, basically it tells you that you might get lucky with much fewer numbers of molecules than in a typical screen against one rigid target. And so that all is buried into the ideas that first we want to do a simple screen, because first in terms of just implementing it, it'll be easier for a complicated assay. Secondly, we want to work with compounds that are drug-like to get to the endpoints faster. But if we fail, we have a backup plan. We can go to much larger numbers of compounds. So that was the thinking. While I'm on this, the activities, activity 1, 2, 3, are roughly prioritized based on what I think is most important. It comes with a budget associated with each one. We were one of the early people to apply for phase 2, so nobody had any idea how the reviewers were going to respond, how the program would respond. So I thought I'd make their life easier by attaching budgets to each of the different types of activity. And if they didn't like point number 9, 10, maybe 8, 7, they could take that away, and I would be dancing and singing if I got the rest of it. So it was set up in a way so that there could be some negotiation over what could be funded.
Great. So just to highlight, so the two main big points from Pradip on this is the first was that when you have your idea of your plan, actually being your hardest critic to say, this is the potential gap, or if this doesn't work, this is my alternative plan. You end up kind of setting yourself up to show, I understand where I'm going, and I realize I don't have the answers to all these parts, but this is what I would do in case it didn't work out. So kind of walk us through that. You've thought through a plan A, a plan B. We're taking your research forward. The next question.
Yeah, a question about the activities again. Is it at all necessary, or did you think about linking the activities to specific individuals within your lab? I'm sure you had that in mind, but was that at all necessary to spell out? You obviously haven't, but is that something that you think is important?
I think linking people, so there is a budget narrative that I also sent to you guys. That's a separate document. If you get a chance, take a look at it. Typically in any kind of funding agency, they want some version of this. So if you propose to do PCR on 5,000 clinical samples, you need people who are good at doing PCR, who are good at data management, who are good at sample allocations and have experience in these areas. In our case, we're doing laboratory-based stuff that involves some technologies that are unique. And so I did mention that John White, who is the world's expert on doing selections against drugs, has already done it before and would be the ideal person to set up this. I have a guy, Joe Smith, who is really great with instrumentation, can make machines talk to each other and so on, so I allocated some time for him. There's a guy called Dan Freeman who is really good at using the statistical programs for looking at relationships between components of genomes that are inherited in specific progeny in a genetic cross, and so he was going to allocate some time for that. I mean, our lab is doing some of these things. Now we're applying all these resources we have to this project. And if you are a smaller lab, you would do the equivalent of this, but find the right partners and bring them in so that you can accomplish what you want.
Thank you, Pradip, for sharing this with us. Really very nicely written. I think I work on malaria, but I found this very nicely, simply written for anybody to understand. I think that was a very big plus point. I just wanted to get a feel from you, since you're so accomplished in getting so many grants, that how much time do you devote on writing a grant? I mean, as for young PIs like myself, how much time do you think, at an average, would be required to put in to write a grant in?
I think it varies. I really am trying to be as honest as I can when I talk to you guys. I think in our business there is a lot of pleasantries. Everybody talks about the ideal world, how things should be. I am terrible about grant writing. I'll be the first to admit. I put things off to the very last minute. I found typos in this as I was reading today, and I was so embarrassed. There's a manual that's been written as a manual, and anyway. I think the most important thing is I really think about projects a lot, more than the writing part. I think you have to know what you're doing and why you're doing it, especially for these kinds of activities where, in phase 1, I don't know what the numbers are these days, but 400 some people apply, and or 4,000 apply and 100 get funded, something like that. You can't take this lightly. You have to be your own self-critic. You have to think about it. You might even play with different versions of this with your own staff or your colleagues, and they are your critics, and they're telling you, watch out for that. Why are you doing this? Why don't you go do it differently? So the thoughts that go into the proposal is not during the writing part. The writing is almost the last thing that I do. But sometimes I underestimate how hard writing is, and I really should devote more time to it. I'm not answering your question directly, but basically I have to run my lab. I have to talk to people in my lab. I have to, when I'm on airplanes, I'm sketching ideas about how to put things together. So by the time I go to write, I'm actually fairly certain. I've written grants of this size in a period of about 2 weeks or something like that, because I have references in front of me in a file. I have thoughts I've sketched out in my mind. And then, as somebody pointed out, it's good to set it aside, go back, revisit it, make sure it says what you're trying to say. This is wonderful. I mean, people are laughing about that video from the Gates Foundation about Grand Challenge explorations, but it is amazing that you can write such small proposals.
Absolutely.
We recently, as you know, got this NIH grant. The size of that proposal was 350 pages.
Absolutely. Yeah.
And we wrote it over a whole summer, and everybody in my lab was assigned tasks to write different parts of it. And there's a completely different way of writing a grant proposal. And when I submitted it, to be honest, I don't know what actually went finally. And I didn't want to look at it anymore, because you just sort of pray to the gods of grant proposals and hope everything goes well.
On this application, you have Dr. Kip Guy. Was he on the phase 1 application?
Who is?
Dr. Kip Guy. I mean,
No.
he wasn't on the phase 1. He's a consultant you brought in on the phase 2.
Yes. So in phase 1, it was all about establishing the concept. So we didn't know that we would be able to do a selection in a 96 well plate. We had reasons to think it should be doable, and so we had proposed to do that in phase 1. And if we got there, then we would go to phase 2. So there was no need for Kip Guy for that first stage. Similarly, we wanted to show that the parents of the original genetic cross in malaria, DD2 and HB3, would show the kind of difference we need in order to be able to dig deep into the progeny. And so we showed that the parents had the appropriate phenotype. There was 10,000-fold difference in frequency for resistance to this compound, which was above and beyond what we would have expected. And so those were the sort of the goals we were trying to hit in phase 1, and that we could do in my own lab.
Okay. I just had a comment which I had required clarification from you. In Pradip's, there's a phase 1 financial and scientific report, and there's the phase 2 application. Now, the basis of the phase 2 application, all the groundwork, the proof of principle comes basically from the phase 1 scientific report. So my question is, will the reviewer get to see all these 9 pages?
Yeah, so the reviewer gets 5 pages of the phase 1 report, then your phase 2 application, and they also get your copy of your phase 1 2-page application. So we see what did you say you would do, what you did, and what are you proposing to do next.
Okay, and just as a comment, I have this phase 2 application template, the most recent that I downloaded from the site. Pradip's application is absolutely in line with this. This does ask for the budget in objectives-based way, and it also, the only thing that he's done here is that in the project plan, he's given his activities based on objectives. So, I think the structure is absolutely
It's been consistent
yes.
through, yes.
I think that guy actually talked about the question I wanted to ask. So I'll ask a different one. How much reference are you allowed to make within the phase 2 application to your scientific report? Or should we just assume, and I think this is already answered unfortunately, that the reviewer will first of all read your scientific report before reading your application?
So I can answer in terms of this, what you'll review, but I think the question about how you reference and what you reference, I think that'd be great for Pradip to comment upon.
So go ahead.
You go ahead.
I just want a quick clarification. Are you talking about referencing your own progress, or are you talking about referencing other people's work, or both?
No, in the beginning I actually thought that you would send an application on its own. But one of the reasons that I was able to understand your application here is because I started by reading your scientific report. If that wasn't the case, perhaps I wouldn't have understood so well what you're trying to apply for. Now, my question was, while you're writing the application, how much reference are you allowed to make to the scientific reports? Especially if you're applying for this before the end of your research, before the end of your phase 1 research grant.
So in terms of the actual review, we have both you get your phase 1 report and your phase 2. So it's the reviewer will read the entire story, so that part can be covered. I think the other component, which Pradip, I'd ask you to comment on, is about references and how you provide references and when you decide to provide references. And how did you think about that in your phase 2 application?
Yeah. So I have a very embarrassing story. You guys think I'm such a great grant writer. I was so deeply engaged in writing the phase 1 the first time around that I actually forgot to reference anything. I didn't reference my own work. I didn't reference, the people in our field. I mean, our work really is new and unique. There is nobody else who does what we do. And yet, it would have been nice to acknowledge some of the other people who work in this area, who have studied chloroquine resistance or something like that. And only after I hit the send button did I realize, because they didn't specifically ask for it, But I think phase 1 was one of those times early on when, I don't know how to describe it. We were all new at this. We were very new at this. And I'm very grateful that it got funded in spite of my big negligence. Phase 2, of course, is much more serious stuff. And we made sure that we acknowledged our own work to show that we have the expertise to do this stuff. We acknowledged some of the early people's work on which we are basing the idea that there are hypermutating parasites, and you've seen the references in there. And we just work within the structure. If the page limits didn't allow for that, I can't remember the details right now, it's been a little while, but we made sure that the references were in there, because now there's much more at stake. I have one other little funny story to share with you. I bet the world is not like it was when I first got my phase 1. I'd never heard anything about what happened to our proposal. And then one day I got a call from the university grants people that basically said, we have a check here for $100,000. We think it might be for you. So this was the very early days of Grand Challenge. I've been told by many people that it's not that simple anymore. People who really know what they're doing are starting to run the show.
Great. Any other questions?
So should I take a few minutes to point out what I think I went out of my way to do for this particular proposal? Because some of the things we're discussing are things you would do for any kind of proposal. Over the years, we've got money from Burroughs Wellcome Fund, from Ellison Medical Foundation, NIH, and so on. And everybody has their own emphasis of what they care about. But grant writing is really about what you heard from the panel today. Yet there are a few things that are unique about Grand Challenge explorations. And so as you write a proposal, you're trying to take into account what you think should definitely be taken into account for things that you worry about. I'm always worried. I'm worried about different things. And so hidden into the proposal are these things to take care of my worries. And today I think I forgot, you're the one who mentioned that reviewers are talking to each other, and some of them are reviewing every line carefully, and they're probably circling certain things that is unexpected because they know the standard stuff that everybody feeds them. And so you want to give them some stuff to circle and bring up if certain points come up. So I have gone in and I've highlighted things for you that I kind of snuck into my proposal because I'm worried about certain things. So I'm going to tell you what I was worried about and why I put those sentences in there. They would normally not be in a normal NIH grant where I know who is on the panel because NIH publishes who the regular reviewers are, and you're basically talking directly to certain people that are on the panel. Here it was blind. I didn't know who it was going to be. I don't know what their expertise is going to be. So I think I'll walk through a few things. Also, I don't know if the instructions say that you should box certain things, you should use red color somewhere, you should use certain fonts, where you should use bold. But I'm using some of these things strategically because I'm trying to categorize different objectives, aims within them, and so on, okay? So you always hear that you don't want to overdo it with colors and so on, but if there are some simple ways in which you can highlight certain things, it makes it easier for the reviewer to track where they are in all your arguments. Other funding agencies that don't allow you to submit PDFs, you can't do this. You send an In the old NIH system, it gets copied by a copier that's been around for 100 years. And as a reviewer, when you see this stuff, you can barely read the letters, leave alone any color, right? Now it's become a little better, but this was a good opportunity to separate things out. So, what we're proposing is that we're going to use parasites in the laboratory that have been isolated from different parts of the world, and we're going to test how quickly they become resistant to some compound that we're challenging them with. We have a rationale for why we pick those compounds, why we think even parasites within a single well can do it. Well, this sounds like a radical, crazy idea, right? That mutations are occurring at 1,000 times higher rate. Because if you learned biology back in 1970s, like I did, biology was about what you learned from E. coli and what you learned from yeast, and that was it. So geneticists tell you mutations occur at a rate of 10 to the minus 10 per base pair per each duplication of a cell. That's dogma. So we're proposing something that might frighten somebody, especially if they're like chemists who are reviewing the grant. So you have to link what you're doing, which might sound risky, to something that they're comfortable or familiar with. And so first, in the context of trying to say that drug developers don't have to worry about this if they're treating heart ailments or neurological problems or even cancer. In cancer, you hear about mutations and drug resistance, but that's within one patient. That resistance doesn't get passed on to the next patient or a community. It stays within the patient. So a very bad drug in cancer chemotherapy can be used on a patient. If it doesn't work, you say to the family, sorry about this, and we're in trouble. And then you turn around and use the compound again on the next patient. And so resistance is an issue in some of these drug development processes, but not the way it is for infectious diseases. And you have to kind of bring that out and show that it's important. Somewhere in there, we also talk about the fact that, I think it was in the earlier summary section, I pointed out that hypermutagenesis is not a new concept. Your cells do this all the time. When you change hypervariable regions in the immunoglobulin gene, there is a machinery that works on that part of the genome. The machinery is triggered in white cells. It goes in, cuts your DNA in 2 pieces. It uses an exonuclease to chew back, and then uses an error-prone polymerase to make many mutations within that region. That's how our immune system can generate enormous diversity to deal with unpredictable things that are coming from outside. You have to link stuff that sounds radical to a malaria pharmacologist to something that they ought to be familiar with, or at least one of the colleagues on the panel is familiar with. So they'll say, oh, this is not so crazy. These kinds of things are known in biology. So I wanted to highlight a couple of those things that are in there to create a kind of a safety net for somebody who might think that this is too crazy. Somebody pointed out that, in the phase 1 award, you're preparing a report. There has to be a clear statement of how you did. We had 2 specific aims in phase 1. For aim 1, we established that the 96-well plate assay is working. So we had to state it very clearly. For phase 2, we showed that a genetic cross can be used to try to find components of the genome that are contributing to hypermutagenesis. So we accomplished both of the things we set out to do. Now, when you analyze this stuff at the next layer, this stuff, as I pointed out, is more complicated. It may lead us to an answer, but it might take many years or maybe a decade to get to the answers by pursuing this route. This was more direct. In phase 1, this was actually a second aim. This was the first aim. But we started playing up the part that we think are the ones to pursue for phase 2. There's a lot of detail. Somebody pointed out that, that we had a lot of detail in it. And that's about establishing credibility. We could have just made a simple table that had all the different clones from the genetic cross. These are the 2 parents of the genetic cross. Which ones can also develop mutations rapidly, which ones cannot. We could have just put the final numbers on how fast mutations are occurring. But I think giving a complete picture of how we do the assay, what are the time frames in terms of how quickly the resistance appears, there are small variations between the different strains. Those are things that to an established investigator add some confidence into how we're doing things. We also laid out a scheme on how we set up the parasites and how we do the assays. Maybe I have some more thoughts, but I think I should pause here and see if there are any more thoughts or comments on what I said. Yes?
So, just wanted to confirm, when you submitted this application, were these things highlighted in yellow? Was that how it was submitted?
No. I just did it on the plane
for
as I
us.
was flying here today. No, I just highlighted those to kind of point out some things that are normally not in a grant proposal. These are little deviations from, but they were there to make a point. And I think everybody's project has some element of that where my advisor used to quote an old organic chemist who used to, some of the greatest scientists, I think, were the ones who discovered all the vitamins. Imagine what it took back in the 40s and 50s to grind up living things or extract things that caused certain ailments and cured certain ailments. Think of all the potential bioassays for vitamin activity. Think of the trace amounts in which these elements are present. Think about what it took before there was NMR, before there was mass spec, to get enough material to actually determine its structure, and then to finally prove that that was the correct structure by synthesizing the molecule and showing that its specific activity was the same as the original isolated material from biology. These were incredible people. And some of my mentors trained with these guys, and they would quote them, and their language is not always socially acceptable today, but they were right about a lot of things. And one of the things was they always kept asking themselves, what is my enemy going to say when I go to a meeting? That's a silly way to say things, but it's true. You're always thinking, what is your worst critic going to say as you present this, and you think it through, and if it is a valid thing that stops a particular approach, so be it. You don't pursue that anymore because it doesn't seem like it's going to pay off, and you come up with alternative approaches that sort of survive your own scrutiny, and that's partly what I'm trying to capture here. And if there are some things that on the surface look like strong criticisms, but you've thought it through and you realize that it's not, then you might as well put in the arguments why you're not worried about that particular aspect up front so that you're helping your reviewers. Any other questions, thoughts?
In your proposal, you've identified a key partner for the compound library, but for perhaps a younger investigator like myself, who's not perhaps quite so well known, how can I actually try and identify potential partners even if I don't actually know them in advance?
Yeah, I think it's good to start with mentors who are older. I'm just giving you my thoughts off the top of my head. I'm probably going to regret half of what I say. But anyway, what I would do is contact people who have traveled widely, have followed people's work for decades, who have seen up-and-coming bright scientists, and seek their advice in terms of who you should approach. And sometimes they can serve as mediators and so on. In your case, young youth. Youth Wong is a great guy to talk to. The reason is, I think, there is the ideal world and then there are the realities. And the realities are that on a phone call or a quick email, everybody's excited about something or the other, or they might be too busy and they don't respond. But even if they do respond, the level of commitment, how much they're going to get engaged in your project can vary. People have different levels of reliability. Some of them, among certain people, I'm considered incredibly unreliable because I won't respond to everything or I don't engage with them on everything that comes through my desk. I pick and choose what's worth my time and energy and so on. So finding the right people is important. And then, of course, they have to be good scientists. I mean, you want people who are really, because when you have complicated data like this, it takes a while to find out whether it's real or not and so on. And so you want people that you're hitching your wagons to that are themselves people that will take you in the right place and not ditch you over a cliff. Uh-oh, what did I say?
Keep going.
Well, so I have a few more highlighted things here from the application for phase 2. And so here we, again, take some sentences from the instructions. And it's again been a little while, but I remember the instructions saying somewhere, what's your idea? What's your goal? How do you consider this in terms of strategic import? Well, take those little phrases from the instructions and make them the headline of your application. It forces you to follow a structure that the funders are familiar with. Makes their job easier. So we're very proud of our idea. And so even though it's been stated before in the summary, it has probably been stated in some form in the phase 1. This is such an important part of the whole application. There's nothing wrong with saying it again a third time. And a good idea can often be presented from slightly different angles. So here's an opportunity to again pull out this idea from a slightly different angle. So we talk in here about a mutosome because what we're trying to convey is this idea that this is not some little polymerase that's just more prone to errors, that creates genetic diversity, that creates resistance. This is a very organized machinery that has to keep the parasite from losing control over its genetic makeup. It only has to kick in when it's really necessary. It has to cause damage in DNA in localized sites and not all over the genome. And people in the field of immunoglobulin hypervariability or adaptive mutations in E. coli have created this idea of a mutosome, which is a complex of proteins and possibly other components that is capable of recognizing parts of DNA and doing the whole process that it takes to create lots of mutations within a region. So we're already invoking an idea into the reviewer's mind that this is not one simple little enzyme. And this is critical for what's coming later. Our odds of being able to block the process improve dramatically if this is a multi-component reaction involving multiple partners. There's no time to explain all this. You use key phrases, you underline them. It's like love. You set it out there and you hope it comes back and helps you. But we do point out that this is similar to what goes on in immunoglobulin genes. So I think this is unique about the Grand Challenge program. They want your wild and crazy ideas, but groundless wild and crazy ideas is like throwing money away, and there are precious reasons not to do that. So you do want to have ideas that are different from what everybody's thinking, that are relevant, but at the same time, the burden is on you to keep showing that this is grounded. Okay, so the goal is to find ways to block this thing. We talk about primary goal. Again, we're emphasizing that the chemical approach is what we're starting to bet on now for phase 2. More emphasis on that. We're not dismissing the biological approach because I told you we actually don't have anything working when phase 2 was submitted in terms of finding a molecule that blocks anything. That's just setting yourself up for trouble. So the biological process, we actually have data of specific loci in the genome that are contributing to a hypermutagenesis. People are comfortable with the idea of chasing loci, finding a target, finding inhibitors. That's a very conventional way of doing things. So we don't want to abandon this in case this doesn't work. Words like malaria elimination is something that's really important. This is what the foundation cares about. And this is what we work on. So it'd be silly not to link your goals with the goals that the foundation cares about. The idea that we're prioritizing things based on direct, practical strategy and not worrying about hypothetical biological schemes that require 100 different things to work correctly before we have a product, that's something that was important and was worth mentioning. And then in my introduction, I mentioned that while we're working on malaria and having a new strategy for making compounds more useful, this is a general new strategy that could be applicable to other pathogenic microorganisms and so on. So that's also worth mentioning under why the whole project is worth funding. Okay? Now clearly, again, none of this was highlighted in yellow. You're counting on the intelligence of your reviewer. They're very good at this kind of stuff. When I'm reviewing grants, when I see things that stand out, I'm constantly circling and marking. I'm hoping that this is what it looks like after the reviewer finishes reading the grant. That's what you're trying to do. So in activity 1, which again I told you is the highest priority for us, we wanted to optimize ways to do a high throughput screen. The fact that we demonstrated in phase 1 that we could do a selection in a single well of a 96 well plate was really important. Without that, there was no such project. But once you've established that, it's still a long ways away to actually getting a high throughput screen running. There are lots of steps involved. And so we talk about some of the potential problems. I'm sorry, my pointer is weak. We're talking about certain potential problems of how you have to change media. Libraries of compounds often have precious amounts of quantities of materials. You can't use the standard methods that we use. So we have to make some judgments on when the exposures would occur to try to block hypermutagenesis. So all this is spelled out in the context of AIM-1, what we still need to do to make the screen work. We had a little bit of a discussion on why we chose to start with 5,000 drug-like compounds. And again, that was to stay close to the aims of the funders as well as our own goals of trying to make things move fast. The quality of these 5,000 drugs makes a difference, and there's some discussion of where we would get those compounds and how we would choose what those 5,000 compounds are. Later on, there's this backup plan where by now, hopefully, we're very good at doing the screens. And at this point, if necessary, we can use larger amounts. If you listen, even though what we've highlighted is 500,000 compounds, nowhere does it say that we would do 500,000 compounds. That's just showing that we have the ability if we need to. And it talks about going in a stepwise manner to approach this process. As with all science, just because you get a hit under certain conditions doesn't mean it's going to be the final compound you want. So there's a lot of validation. I'm sure this is true of all the things you guys do with diagnostics and everything else. You show the most efficient approach to get to an answer, but if you get an answer, you want to have all the traditional ways of validating that the way you approach this was reasonable.
Pradip, one interesting thing to note is you look at all the yellow highlighting. I mean, those are the key phrases that, are to jump out on the page. Many of them are, he has that as activity, and it's that first line. So when you see the subheading or title that you have, that following sentence right after kind of gives you a good sense of where you're going. And then you kind of delve into the details, the plus, the minuses, your plan, but you can get the gist of everything in that first bullet point or first bold point and the subsequent sentence. So keep that in mind.
Yeah, so several people say they like the proposal, and I'm very flattered, but part of that is, I doubt if you actually read every sentence in the grant. What you saw was the skeleton of the thinking just by reading the headings and how it's compartmentalized and arranged in some kind of a logic.
I was just wondering, I didn't receive a copy, I don't know if any of us did, of the budget for this proposal, the second phase. I'm just wondering how, it seems that activity 3 is a mitigation in case you're not able to identify anything in the first 5,000 compounds. But it seems to me the budget would change considerably if you're going from screening 5,000 to 500,000. And how did you incorporate that in the budget?
I think I did it in a time-wise manner. So we had a continuum where don't hold me to this as a legal document of some kind, but basically, I remember, yeah, exactly. That can be edited. Don't distract me. I'm very vulnerable. I already have problems focusing on things. All right, so I think what we did was this is partly why we have these different months in there, is we had a certain number of months to establish an assay, to get going, and so on. And then if this didn't pay off, there were additional months of doing this. Implicit in this is that if the first strategy worked, our energies would go in a different direction, hopefully with the same budget. Nobody was going to take money away if we were successful. Yeah, once you get too involved in this, there are obviously holes in it. But at the first level, it really is as logical as you can get, considering you haven't actually done the experiments yet. Is it okay if I go some? Do you want to stop for questions?
No, I think we'll keep going, and as people have questions, just raise your hand.
And how are we doing on time?
I think we're okay still. A couple more minutes.
Okay. So it's the same thing again. We talked about the chemical component. Now there's a biological component. Again, I'm emphasizing that I realize this is ambitious. I don't want somebody else to tell me that what you're proposing is ambitious. I know it. And if I think it's really scary ambitious, I might have even underlined this. Sometimes ambitious things are worth doing. This is the format in which you're allowed to do those kinds of things. So the biological component is similar to this. The later part of this is about evaluating the value and relevance of what we're proposing. We're really conscious of the fact that we're proposing to do some crazy things. We think it has some value. Here's how we're going to do it in the laboratory. What if we succeed in the laboratory? Wouldn't it be cool if we had a way of taking all these technologies into the field and asking whether this has relevance with other compounds other than our test compounds, with other strains that come from other parts of the world? It's demonstrating that we're thinking way beyond our little lab. We're thinking whether this is going to come closer to where it needs to be implemented. So that's what the last few aims are. And again, they're not throwaway aims, but if somebody didn't have confidence that we were going to get there, I was happy if those budgets were going to be cut and so on. But we would have liked to do the whole thing. All right. And as I pointed out, there were some very key references in our business that refers to people's thoughts on how we do assays, how we think about genetics and genetic change. That was all in there.
Oh.
Okay, I'll stop here.
I had a comment in terms of the activity times that you have put up. Would it have been more useful from a reviewer's point of view to see how they align, in terms of sort of like Gantt chart, and then sort of linking it up to the actual timeline of the project?
Yeah, I think the way the budget was put together in terms of objectives and time and dollars, it was essentially like a Gantt chart. So what I've shown here is the verbal representation of the way the spreadsheet looks. But you're right on target. That's exactly what creates confidence in a reviewer, that you have a rough idea of how long it'll take and whether you will achieve this or not. And every project's different. Some places, you're developing the technology from scratch. Here we had a lot of established technologies that we're applying to a new kind of a question. So we could afford to be a little more ambitious. More is expected of us. So that varies depending on the individual project.
So I guess I just make one or two quick comments. First, thanks. I mean, this has been a brilliant walkthrough, but it's really reinforced a few things for me. And one is taking those few sentences just to kind of reiterate, what might be presumed to be obvious, but doing it in a way that is, really cogent is incredibly valuable, because one of the things as a reviewer is when you kind of get to those last few proposals and you're thinking about which ones you're selecting, you're starting to switch in your own mind thinking about, okay, how am I going to defend this selection? And so having, a really kind of compelling description of how this relates to a strategy, or this is, ties into reasonable things that we know, in your own words is actually incredibly valuable, and it really helps the flow. And then I'd also, really reiterate sort of the structure, because you tend to read the proposal kind of in phases. You get a bunch of them, you scan all of them, you form some pre-opinion, and then you go in and you dive into them in detail, and it's that sort of scan that's very useful as a first framing of how you think about the proposal as you go. But I think this was wonderful. It's a really great discussion.
Thank you.
Let me just use that opportunity to thank Ling and Noel and Pradeep on your behalf for a fantastic after-lunch show. Let's give them a big round of applause.